And now to all who still here after all this very interesting but rather difficult for brain experience and days, this is the greatest pleasure for me to present Professor Victor Tsetlin, who is one of my oldest scientific friends. We are friends for more than 40 years, and who is one of the youngest scientists from the point of view of scientists, because he is one day, when he sees something new, something which is puzzle, he is like a baby actually trying to resolve it immediately, actually just sometimes unpatiently, but very effectively in many things, because he produced a huge amount of papers. Actually, I did have a look in the website of Science, which gives more than 320 publications in very high-ranked journals, and he is extremely creative, he is extremely talented, and person with a huge sense of humor. And I like actually just to interact with him personally and by electronic, because we have always something very funny and very new. So please.
Well, Peter, thank you very much for such generous introduction, but I'm afraid I will become shy and would not be able to give a talk. Okay.
And you have 30 minutes.
Yeah, okay. You know, this line here, the upper line, is not the name of my talk. I just, because being the last speaker here, I want to express my gratitude to organizers for this excellent symposium, and also express thanks to the three ladies who made it to proceed so easily and nice. Marina, Vera, and Alla. So this is fantastic, ladies. And also, I was very lucky that we already heard the talk of Professor Blackburn, because in his first slides, he mentioned Vadim Ivanov. Well, it was quite obvious, because we have the full name of this symposium, but it does not include the line that it does have a tight connection to Vadim Jubilee. So I will tell you that my talk will be connected to Vadim, not in the first two slides, but virtually through the whole 30 or something slides. And why I'm allowed to do this, and well, I should confess here, Peter used the word young in relation to me. It's absolutely not true, because I'm started working my, let's say, scientific or academic career 52 years ago, and my leader was... Yeah. Well, I guess I understand your hints. Thank you very much indeed. Yeah, and I started with working with Vadim Ivanov. And so, not for nothing, in this slide, you see large letters for from and to. And I'll show you that I have to make certain movements in space and time. And I'll illustrate you why these movements occurred. Well, the beginning was 1965. I arrived to this. This is Vadim Ivanov, and I hope it's me. And in space, it all occurred at the Vavilova Street in the Institute for Chemistry of Natural Products, IHPS. And then, as you know, some years later, we moved to this present building, Miklucha-Maklaya Street, 1610. And this was New Guinea. They are not related to our present address, and they deal with the Miklucha-Maklaya. You may find some parallels between these two photos and this, but maybe the rollers are the same. I was at that time Mena as junior research worker. So, and it... Huh? Well, Peter, all the questions later, please. So, and you understand, we moved to the Institute of Bioorganic Chemistry. And my first task from Ivanov was, Victor, you have to synthesize some alanine-containing peptides. Well, at those days, it was not too usual to ask questions, why this or why not that? And only a couple of years later, I understood the reasons. Because just before my arrival, Vadim arrived back to Moscow after spending 1 year in England. And this publication shows what was his activity there. In a colleague of English authors, they were interested in optical rotary dispersion. This method is not used anymore, and so the subjects were 3 and tetrapeptides. But in fact, this was the beginning of Vadim long-lasting interest in conformational studies. And one of the results, Professor Blackburn, who listens to me, already mentioned. It was valinomycin, but this will be in one of the final slides of my talk. Well, you see, some of these papers are ancient, archaic, and are not really visible. But here the list of those peptides which I synthesized. And I'm happy that they have to be useful because they served as a basis, one of the bases for what is NMR known as Karplus-Bystroff curve, because we had a fantastic NMR person in our institute. And I was happy that I was, together with Vadim, a co-author on this paper in tetrahedron in those 69 year. Well, and also in Dr. Friboulet today, I was happy to hear that, well, cyclic peptides, still not an old junk, or should we use more decent words? And next task, which Vadim gave to me, I had to synthesize cyclic peptides of different size. What was behind this? I already mentioned his interest in conformational studies, and he expected, and it happened to be true, that varying the size of the cycle, you change the conformation, and you also could change the types of activity. And in those days, if a naturally occurring peptide was not cleaved by proteolytic enzymes, it was the first hint, oh, this should be cyclic peptides. And many cyclic peptides already were used as inhibitors. But getting pretty large peptides, decapeptides, this reduce and 8-glycine, yes, some of them, smaller one, proved to be inhibitors, as was expected. But when we increased the cycle and increased the conformational mobility, we for the first time got cyclic substrates of chymotrypsin and pepsin, and they were especially and exceptionally of high activity, even much more efficient than linear most popular peptides of those decades. And again, this was published, and today I'm very proud to be one of the co-authors in such a nice company, Ivanov and Ovchinnikov. I cannot hide it. What was the next? And again, in those days, it was a beautiful collaboration of Vladimir Bystroff and Vadim Ivanov. And Bystroff put a task, well, we should have a protein object. What about protein object? And in those days, Yuri Ovchinnikov brought to Institute the idea of protein neurotoxins from snake venoms. And Eugene Grishin was the first to get it in large amounts, this protein from Middle Asian, Uzbekistan cobra, and he was the first to determine the primary structure. And then Bystroff put a suggestion, why would we try to determine the spatial structure by the NMR? Well, listen, those days, 100 megahertz was the best available, and no NMR works over the protein of this sort have been done. Well, and he needed our help. We made selectively labeled derivatives of this protein containing a spin label or some other. And the first, our NMR paper has been published. And we introduced spin labels, determined the distances, and not yet knowing anything about the general shape of the molecule, we're told, well, 2 loops should be in proximity. I don't know how this work would proceed, but 1 year later, 2 excellent groups, one in the United States and one in Great Britain, determined the X-ray structure of similar toxin. You see it here, 3 loops, and the name 3-finger toxins came much later. Well, it didn't mean that we should stop our work on these toxins, and we made a large series of derivatives containing a fluorescent, spin, and later on photoactivatable group in identified position, large series, each time a label of known properties in a particular position. And then we decided, oh, let's try to see how they interact with nicotinic receptors. And I'm happy that we made it. Using using EPR and fluorescence, we for the first time in the world showed which portions of the toxins are in contact with the receptor. Well, toxin is the same shape and type as it's represented today. But here, we depicted receptor as an egg. Why so? But I can tell you, look here, 79. No subunit of the receptor is cloned. No real spatial information is available. And it's over 20 years of fantastic electron microscopist Nigel Unwin resulted finally that 2005 he published cryo-EM structure of nicotinic receptor with a resolution of 4 angstrom. Excellent. Well, but the question might arise, how in those days in the Shemyakin-Ovchinnikov Institute they've got purified nicotinic receptors? Well, the reason is that once Ovchinnikov took me with him to Sweden, and there I met Evert Karlsson, who was a fantastic man in toxins, and by the way, he was the first in the world to purify nicotinic receptor by affinity chromatography on a toxin column. It's true. And so I convinced him to come to our place and to analyze toxin interaction with the receptors. Well, funny enough, when he arrived in Moscow and in our offices, I told, I need a pass for foreigner. They asked me, for whom? And I told, well, Evert Karlsson flew to our institute. And then it was a huge laughter in the whole institute, because in those days, extremely popular was so-called Karlsson the Kid, Malish Karlsson. Well, but anyhow, it worked successfully, and I'm happy that it took place. And also, you see here the people who are excellent working at the institute, like Sasha Arsenyev, Yuri Utkin, Vadim Ivanov, and Sasha Suri. Now it's another place, another institute. Well, so also I'm trying in my talk to depict some achievements we were done with Vadim Ivanov, and later maybe without his help, but just discussing all the data with him. You can ask why bacteriorhodopsin is here. I'm speaking about toxins, receptors, but I told you, I should tell you, in around end 70s, beginning of 80, bacteriorhodopsin and rhodopsin were the most important objects in our institute. It was Yuri Ovchinnikov interest, and it would be reasonable to work on this topic as well. And also I was in Vadim Ivanov. Our contribution was the following. Interesting method of tritium labeling was developed in our country, and we, for the first time, applied this method to the membrane protein, isolated the peptides, accurately determined the amount of included radioactivity, and from this we gave a corrected position of the polypeptide going into the membrane and getting out of it. Well, you can say, well, it's not that interesting. Well, it's not interesting today when every day you can see the published structure of a novel receptor by cryo-electron microscopy. But in those days, it was an important achievement. What next? I already mentioned that in addition to other labels, we started working with photo labels. And I'm happy that our interest met or intersected with these photoactivatable compounds with Ferdinand Hucha, who is sitting now in the first row here and did a lot for this conference. And we decided to combine our efforts. And the result was the following. With one of the derivatives, we identified the cross-link between the toxin and receptor. And this gave very important information that toxin indeed binds between the two subunits at about half height of the ligand binding domain. And it's interesting, and again, science cannot be done all in only one country. Jan Machold made diploma work with Yuri Utkin, and then Yuri Utkin and myself, we were working with Jan Machold for a certain time in Ferdinand Hucha lab, and the result was this publication in PNS, about which I'm still very happy. Well, and now briefly some work, the work on toxins since that years more or less efficiently was going in our lab, and I just show several examples. One class of snake toxins was known at it had the name weak toxins because not much toxicity and their targets were not known. And I'm happy that Yuri Utkin demonstrated that this sort of toxins, they block muscle type and alpha 7 receptors. Please note one thing, and I'll show you later, that classical alpha toxins have additional disulfide here, and this in the N-terminal loop. You will see that's important later. Well, another interesting thing from the snake venom, Urutkin and Alexey Ospov and other colleagues isolated the toxin which was glycosylated. The family of snake toxins is a huge one, but before us, nobody could see the glycosylated toxin, and it may be very interesting and useful because when it's glycosylated, virtually no cytotoxicity. You remove it, and it is as cytotoxic as the initial non-glycosylated compound. Well, proteomic studies are continued today, and mostly because fantastic talents of Yuri Utkin. You see him in his, I would say, office, a lot of tubes, and he has a short angle of his table where he's working at the computer, and he writes paper fantastically now. And of course, it would not be possible if our long-standing collaboration with Vladislav Starkov, who has fantastic and very brave hands. You see it here. Well, some of the results are recently we isolated a novel type of structure. You see these two three-finger toxins connected by intermolecular disulfides. Nothing was found earlier. Interestingly, this modification, natural modification, changed its property. And if the starting monocobra toxin block alpha 7 and muscle receptor, and this dimer acquires a capacity to bind this one. And also one interesting thing that normally in X-ray structures, the conotoxins molecules are oriented this way in opposite direction. And we, for the first time, showed that the structure with such nose-to-nose orientation is also possible. Well, the work on toxins continues, and we are very happy that recently, mostly it was efforts Denis Kudryavtsev, that we discovered that alpha-cobra toxin, a classical blocker of nicotinic receptor, is quite an efficient inhibitor of certain subtypes of GABA receptors. I wouldn't go into details, but I'll tell you that we made some modification, and we've got very interesting information about the binding site of this classical nicotinic receptor blocker at the binding sites of GABA A receptor. The continuation of work on snake venoms and a recently new finding. I see here Yekaterina Wulfus in Pushino, who is for quite some years already in the nicotinic receptor field, and her first publications were in Nature in '65 or '69, and she's fantastically active, and I'm happy that we collaborate with her. And first, with Yuri Utkin, they demonstrated that all phospholipases of snake venom can inhibit nicotinic receptors. And recently, we demonstrated that is also true for mammalian phospholipase. And I wouldn't go into detail, but very important medical conclusions and perspectives can be found, can be realized having this sort of interaction. Well, you know, we have this conference, and it would be impolite to speak only about the proteins, because Vadim is sitting here, did a lot for peptides, peptidomics, and whatever related. So the word peptides is our most favorite word, I should say. And I'll briefly tell you what you've got recently working with peptides. Utkin and Vlad flew to Vietnam. They analyzed snake venom, which was virtually not analyzed previously. I found their linear peptide. What is so exciting about this? But because it doesn't contain disulfide bridges, and really all toxins from animal venoms like snake venom toxins, conotoxins, about which I will tell you later, their structure is always stabilized by one or more disulfides. And this happened to be the efficient blocker of muscle receptor. And we also saw a possibility of this compound as a my relaxant. And in fact, I wouldn't go into details because everybody's awfully tired here. But anyhow, we've got a grant, and its preclinical studies now are almost finished, and we still are full of optimism. Now, in the late, I would say, decade or more, our favorite objects are conotoxin, and I'm happy that the work is done mostly in the lab led by Dr. Kasheverov. And this is not very new results, but I would like to start with this work because I'm happy that at my symposium participated Gus Smith, who discovered acetylcholine binding protein about 15 years ago, and together with crystallographer Tisha Sixma, they determined X-ray structure of this protein. And I approached them, and Igor Kasheverov crystallized the complex of conotoxin with ACGBP, and this was a paper in Nature Structure. What is interesting, today we already heard about this libraries of peptides, and this library of peptides from conus snails, huge. And this was the first example, the first X-ray for the complex made by conotoxin or a cod peptide and its potential biological target. Well, there are some other works, I wouldn't go into detail, but for the first time demonstrated by photo cross-linking that different orientations are possible for the same ligand and binding sites. And a little bit later, with the collaboration with crystallographer for other compounds, we confirmed that this conclusion from the wet biochemistry, from photo labeling, was indeed correct. More recent data. Igor Kasheverov presented a talk at our symposium, and there he told about fantastic collaboration with Roman Yefremov group when they gave us their new invented computer program to design in a better way a new conotoxin. And we followed their advice, and then we analyzed the synthesized compounds by radioligand analysis, electrophoresis, calcium imaging, and we really got several analogs of very high affinity to neuronal nicotinic receptors. While this work is going on, we have collaboration with Chinese people, and recently we published X-ray on another conotoxin, which is very specific for one particular neuronal receptor, alpha-3 beta-2. And I also am happy that a big team 2 years ago, including Chinese people, the leaders, the gurus from Australia and United States, a new conotoxin has been found which has extremely high affinity for alpha-9 receptors. And this is a value because today alpha-9 receptors are considered as a very prospective drug targets against pain. Well, a few words that we work, earlier we were just narrow-sighted and we were working only on proteins and peptides, but we also saw recently that it's much better if your arsenal is more wider. And now we are also working on low molecular weight compounds, in particular in collaboration with Valentin Stonik. This compound was isolated and we found it's very efficient. I wouldn't say very, it's quite reasonable efficiency against alpha-7. It's agonist, and agonists of alpha-7 are considered as potential hints or leads to drug, or not to drug, that's the helping aid for the Alzheimer's disease. And also there was my collaboration with the Italian group from Florence. They discussed which compounds can be synthesized, having better selectivity to one or another receptor subtype. And we did all electrophysiology and calcium imaging, and this is quite good publications in on this topic. Well, now it's almost the final part of my talk. So we are concentrating on three-finger toxins, and now quite a lot of people in this audience already heard from different speakers about LY6 protein, which have three-finger folding. Some of them are water-soluble, and some of them are GPI-bound. And we also were and are working on them. Well, the whole family of LY6 protein is known for many years. But in this year, researchers at the Rockefeller University demonstrated that these proteins having GPI tail, they are colocalized with nicotinic receptors, certain subtypes, and they also modulate their function. Please note that I already showed you this picture, weak toxin having disulfide here, and all this family also has a disulfide, not here but here. So we had a special interest in this molecule. Well, we were lucky that at the department of Mikhail Kirpishnikov, they made the bacterial expression of this protein, and Alexander Arseniev with co-workers, they determined its X-ray structure, NMR structure, and in fact, this was the first well-resolved structure for this sort of proteins. And we also were interested, well, what kind of activity it has. I wouldn't go into detail. We made the radioligand analysis, and our friends, electrophysiologist friends, first in Switzerland, Daniel Bertrand, and later another help came from Marseille, from Peter Brzezinski. So we determined the effects of this protein on the receptor functions. So the main conclusion was the following, that strangely enough, to muscle type receptor, it goes to the classical orthostatic site, it competes with neurotoxins, but with the neuronal receptors, it goes somewhere else, but inhibits them. But since it's not well seen here, but anyhow, since the effect is getting much more stronger when you increase the concentration of acetylcholine, it proves that it binds not in the orthostatic site, but somewhere at allostatic site. But here I should ask the philosophical question, how good are the models? Why? Because what was obtained in E. coli, it was so-called soluble links 1, not containing GPI. But already at that time, the American people published a paper when they used gene with GPI coding something or without it, and the effect on the behavior of mice was different. So the question is, well, how good are the models? Luckily, if we speak about the models of receptors, there are fantastic models like ACGBP or bacterially or expressed ligand binding domains, not bad, quite good, that imitate the spatial structure and functions of the ligand binding domain of the receptor. Well, so I wanted to know what might be here, because earlier in the literature, the proteins, because here another solution is so-called SLARP proteins, which are secreted. So you can forget about the GPI tail. And many versions were available in literature, but they are fantastic, huge fusion proteins, and it's difficult to believe in their activity. And at our institute, the best one obtained at the Kirpishnikov department, but still it contained some addition, N-terminal methionine. And I decided, well, maybe it would be good to get a protein identical in amino acid sequence to the natural protein. And it happened so that I was invited to the very interesting conference named From Venoms to Drugs in Australia. And there I managed to convince excellent Australian chemists, hi guys, why not to try to synthesize this so important protein? And here in this connection, it was really a pleasure to me to go to our archives, and I can tell you the peptide synthesis of protein has been done in Moscow at the Shemyakin Institute in the previous century. Here I'm showing you the afterrefera, the resumes of PhD dissertations from Olga Volpina, 81, and 83, Vladislav Degin. Here you see the sequences, here bungarotoxin, here neurotoxin 2. Here it was the question, how active was this protein? But the polypeptide chain was assembled. Here they got this protein, and Yuri Utkin tested against the nicotinic receptor. We wouldn't tell that it was 100% activity, but it was active. Please know that to synthesize this thing, Olga needed to have 9 fragments and then to combine them. And in this work, it was even 13 fragments. This is the, I would say, a slave of work. And another thing, it was published just in the proceeding of one of the bilateral symposiums. So in a couple of words, so in Australia, they used first two big fragments, then attached to another third fragment, a so-called one-pot native chemical ligation. And they then made the following steps, the, oxidation, refolding. And in fact, they followed the conditions which were early published from the Kirpishnikov department, and they've got good results. And I'm grateful to Zakhar Shenkarev, who gave a table of chemical shifts to them. And we saw that these proteins are quite similar to one another. And that's why, with this similarity of proteins, a couple of minutes more, and not many lectures about after mine, yeah?
Yes, I don't want. I just will sit down.
Okay, so you see that in the paper published a year ago, SLARP1 competes with alpha-bungarotoxin. In our work with the synthetic SLARP, no competition. So what is the reason? And also, if earlier for SLARP, recombinant SLARP, you see the inhibition of current, in our case, with receptors from human receptors on the red receptor, there was no inhibition. But we saw some effect on alpha-7 receptor in the presence of positive allosteric modulator. But still, it was very good that we've got, by synthesis, sufficient amount of this, let's say, long peptide or protein, and we tested them against all possible type of nicotinic receptors, and to our pleasure, we found its inhibitory activity against the alpha-9 receptor, which I already mentioned as the target for anti-pain drugs. And again, the inhibition arises when you increase the acetylcholine concentration. Like in previous cases, to neuronal receptor, the compounds of this family go to some allosteric site. Well, I almost finished with this, and here I would like to stress one thing, that, okay, we see the activity between the early obtained recombinant protein in synthetic one, but I don't want to say anything bad about this protein, because even they have some modified functions, but they seem to be very promising for drug design. But on the other hand, I even have written it, our results clearly show that unraveling true physiologically relevant mechanism for endogenous regulators require the work on compounds which should be as close as possible to the native proteins. I missed this slide, but I wanted to show you because it, in fact, reflects what we are doing today, coming closer from structure to real function of the nicotinic receptor of organisms. But I'm happy that it was presented as a plenary presentation by Irina Shelukina. It has been already presented. Now the end of my talk. This is key people in our department, Yuri Utkin. So we can start with the isolation of the toxin, peptide, and protein from different sources, venoms, and then we can synthesize pretty long peptides, and we can tell them radioligand analysis, immunology, molecular biology, and now she uses a lot on neurobiology, calcium imaging. We have excellent electrophysiologists like Kudryavtsev Denis, and I already mentioned Irina Shelukina, and now the couple of slides for which I really prepared this lecture. One more slide, because people are always finished here showing collaboration all over the world. I missed most of the context, but I was happy that in the symposium which I organized, Gus Smith participated, who discovered this protein. And also I'm proud that I was initiator, and I convinced him to agree to be principal investigator. And we had these grants, NeuroCypress, 15 labs, 6 small companies, and among these collaborators in this grant were people from these European countries. And when I'm showing friends here, it's Koringer and Brzezinski. Well, and now, as I mentioned, we have collaboration with China, USA, and Australia. And now the last two slides for which I made this lecture. Professor Blackburn already mentioned valinomycin, and I want to attract your publication, Ivanov et al. I'm not among this et al. The physical-chemical basis of the functioning of biological membranes, the conformation of valinomycin and its potassium complex in solution. In those days, there were no programs which allow you today to make a beautiful picture of your molecule. Fortunately, my much younger years, I was much interested in painting, drawing, and that's I depicted this molecule and many other peptides. And you see that I was doing it as though already looking at the monument, which you could see. You see the same forest here, as looking to this monument. Well, when you come to our institute, you see this monument. Well, the photo was not done yesterday, it's in winter. But I could not see what is written here. I have a poor eyes now. Okay, I came closer. Oh gosh, what I saw there. The name of this statue is Vadimomycin. And on this, I would like to finish my talk and say, and now I want to say happy birthday to Vadim, but he should work, although his birthday was just several days ago, and let him start the roundtable discussion, Modern Trends in Research on Peptides and Proteins. Thank you.
Okay, thank you very much for this very excited
No
history.
questions, please. No, it's all right. Okay, okay.
You are allowed to have just one question from somebody, if somebody will just will be listening.
I will be ready.
So, okay, so about the maximum response which you have, which you showed for this one, what do you suggest? There is an increase in the response as a maximum response. This is decrease in the expression of the receptor, or is it decrease in the opening of channels, something like that? When you show just with saturated concentration.
Peter, what we were following till now, it's mostly the extent of inhibition of currents. So I cannot, I'm not sure that I was using the word maximal response. Maybe I made it non-intentional.
It was written that was the saturated. Okay, okay,
Yeah.
okay, right. Good. Some other?
Yeah, okay, this is true. The difference between the inhibited and non-inhibited current was most obvious when you increase the concentration of agonist. And it accorded that when we took the same receptor in the cell line, there was no competition for binding of alpha-band neurotoxin, who goes exclusively to the orthostatic site.
Okay, so thank you very much once again. We just oh, there are some other questions? Some comments? Okay, so thank you. Спасибо.